So, thanks for having me here, and welcome. Let's talk about gene expression profiling. Each gene has a different mRNA turnover rate. This means we have mRNAs who have a half-life time ranging from a couple of minutes to even dozens of hours. And this is a problem because let's say we have a gene that is downregulated twofold at a certain time point. And let's also assume that this gene has a turnover rate like the one shown here in red. If we now would conduct a total RNA analysis to measure this twofold downregulation, we actually would have to wait 14 hours to actually measure it. And the reason for that is with the total RNA analysis, we always assume a steady state. And if we're not in it yet, we will always underestimate the actual fold change. However, there's already a solution to that. We can use phothiodine, which is a nucleoside analog for uridine. And when the cell is presented with it, it incorporates the phothiodine in its RNA transcription and as a substitute for uridine at a certain rate. And then we can use the incorporated phothiodine as a tag to biochemically separate new RNA from old RNA, which directly gives us access to the current gene regulation. Unfortunately, this takes up huge amounts of RNA and is also very laborious. Fortunately, in the recent couple of months, there were 3 novel approaches published. I will mainly focus on SLAM-seq today, who overcome these limitations, these disadvantages of the biochemical separation. They all use phothiodine as a molecular marker, but then instead of biochemical separation, they induce a misread of the incorporated phothiodines to a cytosine, which then, when you map the reads against the reference genome, you can then see as T2C mismatches. And as you see, there's no biological separation taking place. So this means now the responsibility for separating new and old RNA lies in our bioinformatic hands because it's done on the data. Unfortunately, it's not as easy as to just take all the reads that have the thymine to cytosine mismatches and count them as new and all the other ones as old, due to a very low incorporation rate of the phothiodine. You can only expect 1 in 15 thymines to actually undergo this conversion from thymine to cytosine. And therefore, if you have 100 base pair reads, you will have over 50% of your actual new reads with no thymine to cytosine conversion at all. Therefore, if you do it the wrong way, you will drastically underestimate the actual proportions of your new RNA. And that's the reason why I'm here, because we wanted to overcome these limitations with our tool globally refined analysis of newly transcribed RNA and decay rates using SLAM-seq, where I encourage you to use the short name Grand SLAM. Grand SLAM employs statistical methods to calculate the ratios from new to total RNAs in a completely quantitative manner, including credible intervals. And today I want to show you how this works and how this performs. So one step back, what are we presented with? In general, we have 2 kinds of reads. We have reads that originate from old RNA, and we have the reads that originate from newly synthesized RNA. Both of them can experience T2C mismatches, because we still have sequencing errors, PCR errors, and SNPs, and so on. However, in the new reads, we have the addition of the phothiodine dependent for T2C conversion. So you can say that the old reads and the new reads experience T2C mismatches with different probabilities. And we can also say that that PC will be greater than PE. Now, to visualize that, we simulated 100,000 reads, each with 10 thymines. We said that around 20% of them should be new, and we chose values for PE and PC, which you can expect in real life. And what you're greeted with are these 2 binomial distributions, one for the old reads and one for the new reads, where most of the reads don't show any T2C conversion at all. Some of them 1, and in this case for the new reads, you have some of them who show 2 conversions, 3, and so on, and 4, and so on. Unfortunately, in real life, you're not presented with such a nicely separated table. and not such a nice separated bar plot. But you rather presented with a mixture of these both binomial distributions. And this is actually what Grand Slam relies on, which is what we call a binomial mixture model. The nice thing about this binomial mixture model is that when we have all the values for PE and PC and so on, we can apply Bayesian inference to estimate the proportions these two binomial distributions take up in this binomial mixture model, which directly translates to the proportion of new to old RNA. Unfortunately, PC and PE differ from experiment to experiment, so we have to estimate them for each experiment. And how we do that is, if you take another close look at this table we just had, you can see that a certain amount of thymine to cytosine mismatches per read, you can be fairly certain that these originate from newly synthesized RNA. So what we did is we took all the reads where we are 99% certain that they originate from newly synthesized RNA, and took the rest of the reads as missing data for an estimation maximization model algorithm, and with that estimated PC. For PE, it was rather simple. All the different mismatch possibilities, they correlate. Therefore, you simply trained a linear regression model to estimate PE. To give you a more picture way, or a short summary of what Grand Slam does is, so you're presented with your whole sample, or the reads of your whole sample, which are mapped against the reference genome. So each read has a certain amount of thymines and a certain amount of thymine to cytosine conversions. We estimate PE and PC for the whole sample, like I just showed you, and then we take the reads for a specific gene. And what we do then is we apply the Bayesian inference model with the values given, and what we are calculating then is the posterior distribution of the proportion values for the two binomial mixture models. And Grand Slam then simply reports the most probable proportion of the two binomial distributions given the data for the genes, or given the reads for the genes. And this is obviously done for all the different genes. So for every gene, you get a proportion value. And the nice thing about the posterior distribution is you get directly immediate access to the credible interval to give you a depiction of the measurement's uncertainty. So much for the theory to Grand Slam. How does it perform? At first, let's have a look at the estimation of PC, which is basically what Grand Slam relies on. So it is important that Grand Slam estimates this correctly. And we simulated data and saw that the deviation is less than 1%. So this is great. But this is not the value you're probably interested in. You're more interested in how well does it perform in estimating the actual proportion values. And again, for that, we simulated data, this time based on real data, where we artificially set the new amount of reads to 50%. And as you can see, the more reads we have, the more precise the estimate of Grand Slam gets. So each dot represents the reported proportion value by Grand Slam, and the bars are the 90% credible intervals. And as you can see, nearly every time the credible interval has the real value in it. So this is what we expected, this is what we wanted. So Grand Slam seems to work. But we didn't only apply it to simulated data, we also applied it to real data. To give you a little background on this experiment, M6A is a cellular mechanism which degrades mRNA at CDS region and 3' regions. And the group from Herzog et al., who published SLAM-seq, did an experiment where they knocked out metal-3, which is an enzyme that does exactly this base modification. Consequently, you expect to get more reads at these regions. And what they did, they used their tool SLAM-dunk, which was published alongside SLAM-seq. And this tool counts all the reads that have T2C mismatches as new reads, and they compared this against the control group. However, with Grand Slam, as we're able to actually calculate proportion values of new to total RNA, we were also able to calculate half-life times out of it. So what we did is we looked at the log2 fold changes of the half-life times of the control group and the knockout group. And as you can see, with Grand Slam, the half-life times calculated from the proportions, we were able to substantially show more differences between the targets and non-targets of metal 3. So to sum this all up, with Grand Slam, we are able to estimate new and total reads ratios from these easily conductible SLAM-seq experiments. And we are able to quantify measurements uncertainty in form of the credible intervals. With the latest update, Grand Slam is also able to incorporate the information from paired end sequencing to give you more precise estimates. And then we thought, okay, we have the SLAM-seq experiments, who doesn't need much RNA, and we have Grand Slam, a tool to leverage everything a bit out of information out of these experiments. So we thought this should be, in theory, applicable to single-cell sequencing. And we didn't stop there. We tried to do it, so we came up with a new sequencing method, which basically applies SLAM-seq to single-cell sequencing, to single cells, which we then creatively called single-cell SLAM-seq. And what we did here is we infected cells with cytomegalovirus. And you can see, so each dot represents a single cell. The red dots are infected cells, the white dots are uninfected cells. We tried to separate uninfected cells from infected cells solely on cellular genes, and we want to do this after 2 hours of infection. And this is a great deal because normally after 2 hours of infection, the heterogeneity between single cells is greater than the differences the infection itself causes. And this you can see is because when we use only the total RNA, that's exactly what I just said, you aren't able to distinguish anything. However, with the SLAM-seq part of this experiment and Grand Slam, the proportion rate is reported by Grand Slam, we are actually able to differentiate between infected and non-infected cells even after 2 hours. So when we realized we have now a temporal resolution of the gene expression in single cells of 2 hours, and if not even shorter, we thought maybe we're also able to show with these experiments the so-called transcriptional bursts. What are transcriptional bursts? Some genes, it's said that some genes do not continuously transcribe, but they are rather transcribing a lots amount of mRNAs in a short amount of time. Then they're completely off for a certain amount of time, and then they're completely on again for a certain amount of time, and so on and so on. And what you can see here, for example, for the well-known housekeeping gene WDR89, that for each cell, we can see it always has some amount of old RNA in it and some amount of new RNA in it. This is what we expect from a housekeeping gene. It transcribes all the time. However, if you now look at the gene HG12, you will see that we only find cells who either have old RNA inside of it or new RNA inside of it. So we are actually able to show these transcriptional bursts with the single-cell SLAM-seq experiment using Grand Slam for the analyzation. And with that, I want to thank mainly my mentor, Florian, who taught me a lot about statistics, and also Lars Dulken, who also helped to bring this paper alive, as well as my lab, the Dulken lab especially, where I want to thank Marissa and Thomas, who did a lot of the wet work for the single-cell SLAM-seq stuff, as well as the guys from the Helmholtz Institute and Core Unit Systems method of Würzburg, who also are involved in the single-cell sequencing, as well as the funding, and of course the ISMB and the RNA Society for granting me the travel fellowship, as well as the graduate school from Würzburg, who also helped me financially with that travel fellowship to come here. And of course I want to thank you for your attention. And if you have any deep questions, like, for example, the statistics, the pure statistics behind all that, I encourage you to come to my poster at 8:30 tonight at 6 p.m. Thank you.
Hi, Very interesting talk. It seemed like you benefit from more reads within the gene. Do you then have trouble with short genes as opposed to longer genes?
No, basically it doesn't matter how long or short the genes are, as long as they are covered with a lot of reads.
But if there's fewer places where you could get that transition of the T.
Yeah, but the transition, I mean, you have. Oh, you mean in that case?
Because there's fewer instances of them in the gene itself. So like with something like Titan, where it's absurdly long.
Then in that case, it depends how strongly this gene is transcribed. I mean, if there are a lot of transcripts inside of that, you have a lot of transcripts where this could happen. If it's lowly transcribed and short, then yes, this could be obviously a problem. Yeah.
Just curious.
Yes.
Nice talk. So in your graphs, when you compare to the other method, the dunk, slam dunk, yeah.
You mean?
So I was just curious.
That one?
Yeah. So some of those graphs don't look so great, right? Do you want to comment on that?
Oh, they are great, in fact, because.
Okay. In the eye of the beholder.
It's what we expect. I didn't go into detail because of the time.
Yeah, that's why I was hoping you can explain. Yeah.
You don't expect to see or to be able to measure these M6A base modification at the 5' region, because there doesn't happen. And this just shows you that, well, we can't actually. This is more like this. This is just random because you shouldn't be able to measure anything there. So, as I said, intentionally. Yeah. Well, thank you.